students, welcome to Biomics Hub. I hope you all are familiar with a small term that is stomata. Stomata is present inside the plant and it is required during the process of transpiration. That means water loss from the plant. So what happens when the plant is in the drought condition? How this transpiration is being controlled? This transpiration is controlled by closing the stomata. When the stomata is closed, it has a less chance to help in the process of transpiration. So how it is being controlled? The plant does not have any brain, so how this opening as well as closing of stomata, whether the process of transpiration will take place or it will be inhibited, how it is being controlled without any brain? So the answer is, it is by the abscisic acid. The plants are having phytohormones. One of the phytohormone is abscisic acid, who is controlling the stomatal closing. So let us understand how the process of stomatal closing is taking place in presence of the phytohormone abscisic acid during the drought condition. What happens during the drought condition? There is synthesis of abscisic acid in the plant, and it is controlling the formation of, or we can tell that it is controlling the stomatal closure. Here we can see that here the abscisic acid is present. When the abscisic acid is present, it comes and binds with its receptor, which is known as RCAR, PYR and PYL. Before going in depth, we have to understand what are the components it is present in this guard shell. So in this guard shell, there are K plus channel, K plus influx, potassium influx channel. This potassium is having both the influx as well as efflux channel. Here we can see that here it is having the efflux channel. Here we can see that here there is a channel for anion efflux. This is the anion efflux channel. As well as it is having the influx channel for Ca2 plus for calcium. Here we can see there is a H plus ATPase channel. This is present in the guard shell. Okay. Here what happens, there is a presence of vacuoles, and in this vacuoles also there is present of these channels like K plus efflux channel, A minus, or we can tell that anion minus efflux channel, as well as calcium efflux channel. So there is formation of much solutes over here. Okay, so we will understand how this abscisic acid is controlling this. Abscisic acid, it is binding with its receptor and it is inhibiting the PP2C. So how the signaling is taking place already we have discussed in our last video. If you have not watched that video, I will provide this link in the description box. Please do watch that first, then come to this video. So this PP2C, what is this? It was inhibiting the OST1, which is a NADH oxidase. When this is being inhibited by the receptor, this PP2C, which was a phosphatase, it is being inhibited by this receptor. Now it cannot inhibit the OST1. The work of this OST1 is that it will activate RBOH, which will help in the production of some of the ROS. Due to this ROS, that is H2O2 and O2, it will activate the calcium influx channel, due to which there is input of calcium inside the guard shell. Okay, this is one way how the calcium is being transferred inside the guard shell. So here, now the work of this calcium, and this is one way how the calcium is being transported. Now the other way, this PP2C which was present, what it was doing, it was inhibiting the calcium ions. By default there will be some calcium ions present in the guard shell. So this calcium was inhibited by the PP2C. Now the PP2C is being inhibited by this receptor. So now the PP2C cannot inhibit the calcium ions. Due to this calcium ions, there was in this vacuoles, there was PLC present. This PLC is being converted into IP3, and this was interacting with this calcium channel in this vacuole, and it was helping this calcium to export from this vacuoles. This is the other way how the calcium is being transported to this guard shell. This is the second way how the calcium is being transported inside the guard shell. Okay, this calcium which is present, it can also activate the CPase, which will help in the activation of this ion channel, by which the anion will be transported out. So by default, this work of this PP2C was also, it was inhibiting this anion channel, but in presence of this calcium ion, this anion channel will be activated, and there will be out of this this anions which are present, it will be transferred out. The here main anions is the chlorine, Cl minus. Okay, so we have understood how the process, how in what mechanism the calcium is being transferred inside the shell. Okay, so what is the next work of this calcium which is present? This calcium which is present, what does it? It also interact with the K+ influx channel. So what is the work of this K+ influx channel? That it will transfer the K+ from outside to inside. So this calcium, what it does? It inhibits this channel, due to which there will be no influx of this K+ channel. Okay, so here the K+ is more outside the cell. Okay, so what happens when the in presence of this abscisic acid, this PP2C is being deactivated. So as it is deactivated, it cannot inhibit this work of this calcium channel. So work of this calcium channel was that it was inhibiting this. So now it will be inhibited and there will be no influx of the K+ ions. Okay. Now next, what is the work of this calcium? The next work is that it will also it will come and bind with this channel. It will help the CPKs and it will interact with this anion channel and the efflux of the calcium anions will be done. So the Cl- is also transferred out from the cell. Here there is no influx of K+ channel and there is efflux of chlorine channel. Okay, so the other work of this calcium is it interacts with the proton pump and it inhibits this proton pump, due to which the H+ which is present inside the cell, it cannot be transferred out. Due to which the pH increases. So here what happens? It is acidic. It is acidic condition inside, and outside it is alkaline. So what happens here? Here the condition is alkaline. Here K+ is present in more amount. Here the chlorine is present in more amount. Okay, so due to which there is osmotic disbalance. So what happens? Here the water H2O which is present in the guard shell is more now as compared to the outside environment. So the water will be transferred from outside to inside, or from low solute concentration to high solute concentration, the water will be transferred. In this way the water are being transferred outside the cell, okay, to maintain the osmotic balance. So during this closure or when the H2O is being transferred out from the guard shell, there is flaccid condition in the guard shell, due to which there is closure of the guard shell. Okay, I hope you have understood. So here there are different type of ion channels which are present, and the calcium is regulating more in controlling this ion channel for closing the stomata. So what happens? There is abscisic acid which is synthesized during the drought condition. Abscisic acid comes and binds with the receptor. It controls the downstream pathway. Like there will be more ions which are present outside the cell, due to which the amount of water will be more inside the guard shell. So as the amount of water is present more in the guard shell, it needs to be transferred out, due to which the guard shell will be flaccid, and due to the flaccid condition of the guard shell, the stomata will be closed. In this way the abscisic acid is regulating the closure of the stomata, due to which there will be no water loss during the drought condition. I hope you have understood this video clearly, understood this topic clearly, and if you are having any doubt, please do ask me in the comment section. Thank you everyone for watching this video. Thank you.